Tepotinib on the mutants D1228V and Y1230C using virtual screening. So here the mutants are D1228V and Y1230C. The targeted gene is MET and the PDB is 4R1V. So the methods we used are the database search, which collects the structure and organized data that is stored in electrically protein structure properties. The protein is characteristics of amino acid and forms a skeleton model. The homologen modeling is computational structure prediction method. Docking, it's basically the binding sites it gives you. And then the virtual screening is techniques for drug discovery. So the purpose is to help make new treatments for this non-small cell lung cancer for patients with these mutations. To determine the effects of the FDA drug to see the outcome of the kinase activity, the mutants will be tested with the FDA drug and the wild type and compare the results to them with also the zinc compounds that are also being used. And to even further the study, the FDA drug could even be placed inside these cells with the mutants and wild type to see the outcome. So here, the docking scores. As you can see, the better docking score is this mutant right here, D1228V, because it's more negative, which means it's going to have a better binding energy, which means it's going to be more stable in the protein ligand complex as well. But the other mutant isn't as, it's still a good mutant, but this one's going to be better used with binding to the FDA drug here. And here's just the workflow of the virtual screening and how it's being prepped and everything. This is going to help predict the binding affinity with the mutants of the wild type and the stability. The modeling helps give insight in the structures and coordination to help create mutations. And this is just a table of like the expected results. You're going to see that the wild type should be greater than the mutant, which will be going in the right direction with the FDA drug, but that cannot also. The FDA drug might not be the best, which could help lead to testing on a different drug as well. That's it. What's it treating? Non-small lung cancer. Non-small cell lung cancer.
Okay, so you actually had all the components for A, but I want you to rephrase some of the things so it makes kind of more clear.
Okay.
So you have those IC50, which are the kinase assay. So that's basically in your workflow, you have adding last step, which is the kinase assay. That's the wet lab. And then you go to your title, you also want plus virtual screening plus the kinase inclusion assay.
Okay.
Yeah, just as it. You want to indicate you also do the wet lab kind of.
Okay.
Okay, good.